Everyone. My name is Jon, and I'm actually one of the new co-hosts of the CGDG event. I'm a postdoc here at the Roslin Institute in Plant Breeding and Genetics. And for today's talk, we have Lingzhao Fang, and he will be talking about farm animal genotype tissue expression, multi-tissue atlas of regulatory variants in farm animals. Lingzhao obtained his master's degree in animal genetics and breeding in China. He then moved to complete a sandwich PhD in quantitative genetics between Aarhus University and China Agricultural University. He then moved to the US to do a postdoc in bioinformatics and quantitative genetics in USDA and University of Maryland. And after that, he joined Albert Nessa here at the University of Edinburgh, where he still works as a research fellow, but now at the MRC Human Genetics Unit. So if you're ready, you're very welcome to start.
Yeah, thank you very much for the introduction. Yeah, I just start to share my screen. Yeah, I hope it works well. Is that okay right now?
Perfect.
Okay, great. Cool. So I'm already started. So, yeah, thank you again for the invitation and the introduction about myself. And today I'm going to talk about some results from this project, the international collaboration. So trying to understand the genetic variants that are associated with gene expression across multiple tissues in the farm animal species. And so this is just the institute that got involved in this in terms of data contribution or data analysis. And so this is just some background about why we need to consider the molecular phenotypes. And this is just from integrative genomics to understand the genetics of the complex traits. So we know the different kinds of genetic variants in the genome, and right now we're focused on SNP in today's talk. So when the SNP happens, we know that SNP probably regulated this intermediate molecular phenotypes, and then all the way and to associated or maybe the causal factors for the complex traits. So in the past almost 20 years, even in livestock, more than 20 years ago, we started to look at the genetic variants and that's associated with the complex phenotypes, both using linkage mapping or GWAS recently, or some population genomics. So if we want to understand the molecular mechanisms underlying each of these associations, we do need to collect all these intermolecular phenotypes. In humans, they'll do a lot of international collaborations and collaborative projects has been launched, and they got a lot of very interesting findings based on this data. So this is also a very big challenge because all this functional notation, functional data are very context specific. They could be different across tissues, developmental or diseases. But once we got this data, this rich data, and we got more opportunities and to develop advanced methods and to understand the circuitry of this, the regulator circuitry underlying the complex traits in human and livestock or plants. And after we understand it, we'll have more opportunity or many ways than to manipulate this circuitry. And so the much closer to the precision medicine or in animals could be plants is more likely to be a precision breeding. So this is the human GTEx project. So this project, their targets on transcriptome, one very important intermediate phenotypes. This was started more than 10 years ago. So it's a very successful project and impactful project in human genetics. So they collected the transcription and whole genome sequence data from 50 major tissues across the human body and from about 1,000 individuals. So they take the gene expression, the average gene expression, and also alternative splicing patterns as the major molecular phenotypes, and to look at the genetic determinants of these phenotypes. And just at the beginning of 2018, when I did my postdoc in USDA at that time, and this is also the major term of the human GTEx, we started thinking about, can we do the same thing and develop the same resources for farm animal species, because we're also quite interested in understanding the genetics of the complex traits, of very economic value in the livestock. But if we develop the similar project like human, it may take a lot of time and a lot of money. So probably it's not possible at that time, and to have a grant to support that. So we started and to look at the public data, and from the public data we also can collect a lot of the RNA-seq from different tissues. And by using this, we can, open this black box between the association with the complex phenotypes. And we finished the cattle. So today I'm not talking a lot about the cattle. And this paper already put the Bell archive, so you can easily download them and read them. And the paper was accepted in Nature Genetics recently, so it will be online very soon. And today I'll focus on pig. We're also working on chicken, sheep, and goat. But the pig are almost finished all the analysis. So the results will be released in a few months. Hope that will be helpful for lots of people working in this area. So the farm animal GTEx and for actually the pig GTEx project recently is a pilot phase. In the pilot phase, we are targeting on the public available RNA-seq data. And so that started more than 2 years ago. And so the expected deadline just in several months. So this is the data summary and what kind of data and how many samples have been analyzed for each of these data types in the pig GTEx project. That including over 10,000 RNA-seq samples and also 1,600 whole genome sequencing data. And this data were used to build the Genotype imputation reference panel. It's quite similar to 1,000 genome project in human and also 1,000 bull project in cattle. And we also uniformly analyze the public available, already published, the epigenetic data, also single-cell RNA-seq data in pigs. That data were used to predict the chromatin states to detect the cancer promoter in multiple tissues. And we also analyze the whole genome by sulfur sequencing, this is DNA methylation data, and also high-C data to detect the TAD. And all this, also the single-cell RNA-seq data, all these multi-omics data were used to note the eQTLs we detected in this by combining the RNA-seq and the whole genome sequence data. So at the end, we also collected the GWAS populations, and to do, we imputed the genotypes to the sequence level using the same panel. And so at the end, we got the GWAS summary statistics for over 260 complex traits. And we also have some validation populations just to validate the eQTLs, and also some validation to validate the GWAS. For the validation populations for eQTLs, and we need the population have both RNA-seq data and also genotype data. That could be from the SNP array or whole genome sequence from different tissues. So that's just some examples up here. Let's first look at the quality of this data. So that a lot of students got involved the data cleaning, because there are a lot of totally mice when you start to look at the public available data, because they were uploaded by different groups and need to go to retrieve all these metadata table and to clean them as better as you can. But after analyzing all this data, use a uniform pipeline and remove the low quality data size. So we started to do some clustering of the samples based on two important molecular phenotypes, like gene expression and alternative splicing. And we can see the samples could be clustered quite well according to their tissue types, no matter they're from, even they are from different labs around the world. The samples can still reflect the tissue specificity. And this also allows us to do eQTLs within each of the tissue to start to look at the tissue-specific gene regulation. And the same panel, including two plots, the left one, and this is the sample cluster based on the genotypes from the reference panel. This is the whole genome sequence data. And this is the samples from RNA-seq population and also cluster based on the genotypes derived from RNA-seq. And in general, you can say these two populations have a similar population structure. And that's also a good evidence that we probably can do a better or maybe a good enough imputation by using this reference panel and to impute these targets RNA-seq population. And then another section, and so also the first output about this project is the gene expression atlas for in pigs. And because this is the largest gene expression data size, including over 100 different tissues and cell types. So by looking at this data size, we can detect some tissue-specific genes, and that actually the gene was specifically highly expressed in a particular tissue. And then we can look at the function of these genes, and you can see these genes, the function of these genes can clearly reflect the tissue biology. And this is also another evidence to show that public data can give some knowledge about the tissue biology. And another very important application for this large expression atlas to better understand the function of the genes, because in the current pig genome, there are lots of genes. We only can say they have the ensemble ID, but we don't know without annotation. So because we have multiple, we have many individuals in each of these tissue, so we can do some co-expression analysis both within tissue or across tissue, and to assign these genes without annotation information to some genes already with annotation. So after that, we can better note over 13% of the genes based on co-expression network. So we use the 5 different distinct cluster methods. And the most important part about the PigGTEx project is called molecular QTL mapping, just to try to detect the genetic variants that are associated with the molecular phenotypes, especially for gene expression and alternative splicing. So the first step, lots of the RNA-seq data in the public domain, you only have RNA-seq data without any genotype data in the same individual. So we need to, we need to call genotypes, the SNPs from RNA-seq. And then we also need to impute that SNPs to the sequence level. So we do this and very carefully and and then check the different, use a very stringent cutoff, because when you call SNPs from RNA-seq, they have a lot of things and can give you a false positive and SNP calling. After this, at the end, we show we can do a good job and for over 3 million SNPs. And then we validated this imputation for just this focus on these 3 million SNPs. So the first validation strategy is we got some individuals, we have both RNA-seq and whole genome sequence data, and this is from different breeds. So we impute the genotypes from RNA-seq only, and then compared with that loci, the matched loci that directly called from a whole genome sequence data. And then we start to look at the concordant rate. So the average is just for these 3 million SNPs is over 0.97. And another strategy is that in some populations, we have both RNA-seq and 50 maybe SNP array, that could be a 50K SNP array. So we impute the genotypes that are called from RNA-seq only, and also compared with the genotypes imputed from this 50 SNP array. And so the different populations, but at the end, we can see the concordant rates over 0.95. So all these validations show us these 3 million SNPs probably is good for the downstream analysis. So this is the distribution of SNPs across the chromosome. As already mentioned that the samples can reflect the tissue-specific function. And so we think it's good to look at this gene regulation or genes or the expression within each of the tissue. So then the first step, because we need to detect if any samples within, for example, in liver, are they from the same individual? Because a more extremely situation is that, for example, all 500 liver samples just come from one individual. So that will be a big issue for doing this population analysis. So by looking at that, we built the IBS and also GRM metrics using the independent SNPs. We got a population, we know that samples are from the same individual. And then by looking at that sample, we at the end we decide to use this 0.9 as a cutoff and to detect the duplicated individuals within each of the tissues, and then keep the one with a higher quality for the downstream analysis. So after that, and we got 34 tissues or tissue groups, and with over 40 individuals. And so we think the 40 individual probably is a good sample size for conduct eQTL analysis. Although, later we'll see it's not that good, but it still can give you some significant signals. So, and within each of the tissue, corrected for the population structure, and also all the unknown and confounding factors by training the peer factors. And I think that we started to mapping the QTL, the cis-eQTLs for protein coding genes. And so the B panel is just to showing and how the sample size are related to the eQTL detection power. So the x-axis is the sample size for each of the tissue. The y-axis is the number of E genes, the gene with a significant eQTL, divided by the number of tested genes in each of the tissue. So in general, you can say there are good correlation between these. So that also means a larger sample size give you more power and to detect if a gene is with a significant SNP or not. And then we start to look at can we detect some independent signals for each of the gene? In other words, that if any gene have more than one independent QTLs that's associated with its expression. So that's we indicated as 1, 2, 3, or less than 4 probably. So the sample, the tissues were ordered based on the sample size from the smallest one to the largest one. So the largest one is muscle. And the red dot is just the mean number of the independent eQTLs per gene. So in general, you can say when you have a larger sample size, you got more power to detect the more independent eQTLs per gene. That's also another signal here, very important thing to know that when you increase sample size, you may detect almost all the genes are E genes. At some point, for example, maybe you have 1,000, but you can't detect all the independent eQTLs for each of the gene because some of the eQTL, especially for the third and the first eQTLs are with very small effects. So you need a much larger to detect them. And then we also compared the commonly used linear regression and with the GRM, the method commonly used in livestock species. And so we look at the correlation between the effect size and the p-values, the significant level, but in general, they are very significant correlated, highly correlated. And the linear, so we in the linear regression, we control the population structure based on the PC by different genotype PCs. And then after that, we start to use the different strategies to validate this eQTL. So because this is from the public data, there are a lot of confounding things can give us the false positives. So we need to show that this eQTL are really good for the downstream analysis. The first validation called internal validation. So we got the tissues where we have over 200 samples. So we randomly divided these samples into two groups and then perform cis-eQTL mapping within each of the group and then validated in another group. And we calculated the pi1 statistics and to measure this replication rate. And in general, these values are ranging from 0 to 1. If marked close to 1, that means you did a good job. So in general, across all this tissue, you can see these numbers are quite large, close to 1, as in general, to show we did a good job. At least for internal validation, especially when the tissues have a larger sample size, like muscle. And the next step, we start to looking at the data we never touched before. That's called the external validation. And the first data size is the RNA. This is the population, including 200 individuals. And they collected the RNA-seq from whole blood cells at 2 time points that denoted as T1 or T2. And besides the RNA-seq, they also have the genotypes, the 50K genotypes from the same individual. And so that's what we know as RNA-seq and the SNP chip because we impute the genotypes to sequence level only from RNA-seq or only from SNP 50K SNP array. So at the end, we got 4 data sets and also this discovery population we have. And we start look at the replication between each of these pairs. And so on average, you can see we got very good replication rate is over 9.5. So I think we start to look at are these eQTLs have the same direction of the effect size and the effects. So by looking at this is the correlation. For this score, the effect size of eQTLs in general can see they are positively correlated across all these data sets. And then we also look at the external validation for another 3 tissues, including muscle, liver, and one of the intestine. And so in general, we got the same thing. So because the left column is the discovery population. For muscle, we have over 1,000 individuals compared to 100 individuals in the validation population. So it's a good thing that in this validation population, they have both RNA-seq and the whole genome sequence data. So we don't need to do any imputation anymore. So we can directly get genotypes from a whole genome sequence. And then we start to look at the pi1 statistics. So we can replicate over 99% of the eQTLs detected in this external validation, but they can replicate it 70%. The same trends in liver but the opposite. Because for this in testing, because we only have half the data size like they have the 100 individuals. So that's a replication or directly related to the sample size you have. And then we also do another complementary analysis for cis-eQTLs. Because cis-eQTL analysis, the population analysis, there are a lot of confounding factors could be an issue for population analysis, like the population structure. But AICE, allele specific expression, is the analysis at the individual level. So that confounding factors is not a big problem. So we do this, after we do the allele specific expression, and we look at the matched loci between these two analyses and look at the effect size. And we can see there are good correlation. And this is just the two examples in muscle and brain, but this consistent across all the tissues we have. And this correlation is quite good and is almost the same as human GTEx detected. So at this point, we think that the eQTLs probably is good, is maybe a good resources for the downstream analysis. And we also do the QTL mapping to another 4 different molecular phenotypes. The first one called splicing QTL, and this for link RNA gene expression and for individual exon expression, and also for the enhancer region expression. And another thing that we need to ask how this eQTL was shared across breeds, because in the previous analysis, we put the samples from different breeds together and to increase the detection power. And then we need to show that the breeds to share the eQTLs. And so we take muscle and liver as example. Here I only show the results for muscle. So in total, we got over 1,300 muscle samples, and this will come from 8 different ancestry groups. That's including 4 crossbred. That's including like Landrace and Yorkshire, which has the largest sample size, and also have some 2 commercial breeds, and another we put all the Asia pigs together as called the Asia breeds. And then we conduct the cis-eQTLs within each of these 8 groups, and then look at the replication rate between these groups. And we can see the Landrace and Yorkshire, like I mentioned before, the sample size is the key factor that related to the validation, to replication. So this large sample size can replicate it over 95% of the cis-eQTL detecting other breeds. And even for Landrace, which have less than 50 samples, can replicate it half of the cis-eQTLs detecting in other breeds. And then we start to look at the correlation of the cis-eQTL effect. And you can see the eQTLs detected in the muscle from different ancestry groups. They will cluster quite well according to each other and positively correlated and clearly separated from the eQTLs detected in other tissues. And the meta-analysis across breeds also give a clearly signal. That's you can see these are highly correlated, the file size derived from the meta and with those derived from the combined population. So this is also a good signal to show that the eQTLs, the majority of the eQTLs, the cis-eQTLs actually, and were shared across breeds. But this correlation not exactly to 1, and we also detected the breeds interaction QTLs in our analysis, but I didn't show here. And the next step, we start to ask how this eQTL shared across tissues, because this is a very important question. And when we started to generate it in the next step, generate eQTLs in tissues, but we have limited bodies, and which tissue is more informative, and we need to consider about that. And then we cluster the tissues based on the effect size of the cis-eQTLs. How in general, you can say the tissues with similar function are clustered together, like all the gut tissues will cluster here, and the brain regions cluster here, and the immune tissues will cluster together. And so here the y-axis is this is just the distribution of each of the particular tissue and the correlation with all other tissues. And then in general, you can say for most of the tissue, they can replicate the correlation is a half. So that they do share a lot of these cis-eQTLs, but there are some tissue was very distinct with each other, with others, like testes. So it's quite different. And because the testes have a very distinct transcriptome profile as the other tissues, also the similar finding in humans. And also the early development, the tissues from the early development stages are quite different from other tissues, at the adult age. And then we also do some another additional analysis and to look at how these 5 different kinds of QTLs were shared across the tissues. And this is 1 to 5 means this QTL only detected in 1 to 5 tissues. And this the last column is means the eQTL shared across all the tissues being tested in this project. So let's focus on protein coding genes, eQTLs, and we can see the red bar. They just show this U shape that could be have a large proportion with tissue specific, but they also have a larger proportion that was shared across all the tissues. But for other types of the QTLs are more likely to be tissue specific, especially for exon or splicing QTLs, also Lingzhao eQTLs. And so another very interesting finding that we started to ask the question, any of the SNPs can have the opposite effect for the same gene between tissues. This is just one example, but in total we detected 600 of the genes have this behavior. This is the gene name. And by looking at effect size from the cis-eQTLs, you can see the same SNPs almost in 100 in RD, even not so different SNPs, but they are in completed RD. So they have a negative effect for this gene expression in testes, but have a positive one in blood. That means the SNP when they increase the gene expression in blood will decrease the same gene, the expression in testes. And the next step, we start to look at the functions of these molecular QTLs. By integrating with omics data, I already show you, we uniformly analyze all the public omics data in pigs so far. And the first is called sequence ontology. And this is just based on the sequence features that you can see the 3' or 5' UTR, and the missense variance, that's nominal variance, and also moderate, low, and high. That means if the genetic variance have a big impact on the sequence, the protein sequence. And so in general, you can see all these 5 different kinds of the QTLs are enriched in moderate variance and the low variance. They do not have a very high impact on the sequence. And they're also enriched with the missense, especially for splicing QTLs. And also 3 and 5' UTR region. And for focus on the splicing region, we can see a much higher enrichment for splicing QTLs compared to other kinds of QTLs. And also splicing QTLs have a higher impact and also enriched in the splicing acceptor. But for the splicing notation, are quite poor in the current genome because you can see that the standard error is quite large, but you still can see the significant enrichment. And the x-axis is log transformed, so the very good enrichment here. And the next step, we start to look at the chromatin states. And the chromatin state were predicted by the combinations of the epigenetic marks. So we predict like a promoter region or some regions were quite close and to TSS, and also the different kind of enhancer regions like a strong enhancer, a weak enhancer, or medium enhancer, or some post enhancer, or will predict the toxic islands. And also that just means only have a toxic signal over there, and also some bivalent promoters or the repressed polycombo. In general, you can say these QTLs have a very high enrichment in the active promoter, and then also some chromatin states close to TSS. But in general, we do not see a very good enrichment for enhancer, and we do see a depletion from this repressed polycombo for these QTLs. And the next step, we start to ask why they didn't have a very good enrichment in enhancer. And so, because we know the enhancer show a very strong tissue specific activity, and we start to look at, say, if this enrichment show a very good tissue specific pattern. So because in all these tissues, we both have this their enhancer notation for these 5 different kinds of enhancer, and also have these cis-eQTLs. And so we started to look at the enrichment. So the blue dots is just means the matched. So that's for example, this means the adipose enhancer was enriched with adipose cis-eQTLs. And so in general, you can see the blue dots was just in the outside of the box of distribution. So that's a good evidence to show that this cis-eQTL enrichment and show a very with the enhancer regions show a very tissue specific pattern. And the next, we start to look at the fine mapped QTLs, because you know the first fine mapped SNPs always have the largest effect, and the second, the third, they may start to have small effects. And then we found the SNPs with the largest effect on the gene expression, they more likely enriched with the active promoters compared to enhancer. But the SNPs with small effects are more likely to enrich with enhancer compared to promoter. The E panel, so we also look at the DNA methylation data, because we detected this the tissue specific hypomethylated region is the region with tissue specific low methylation, because that region are more easily to be open region and were bound by the transcriptional factors. In general, the same pattern we observed. The blue dot means the matched tissue. We can see a very good enrichment for the matched tissue, especially for liver, and also some blood. And within muscle, we got over 100 samples, and with DNA methylation data. So we conducted the methylation QTL within muscle, and then do the colocalization between methylation QTLs and with all these eQTLs. We do see a lot of methylation QTLs were colocalized with the eQTLs. So I didn't show the result here. And the G panel is we'll start to look at the high-C data, and we would like to ask one question: if the cis-eQTLs are more likely to be located within the same TAD region and with their target genes. But if you consider the TAD, you need to think about the distance, because if the SNPs are more closer to their target genes, they have a larger chance to be within the same TAD. So after carefully taking account of the distance, we start to look at the enrichment for the long distance of cis-eQTLs with their target genes. And so for example, this is the upstream and this is the downstream. So it's the over, if the cis-eQTL is over 400 kilobase pairs away with their target genes, and then you can start to see the significant enrichment. And the H panel is just one example. So this is the high-C contact plot, and this is the target gene. This is called BAT23, and there are two eQTLs, independent eQTLs. If you look at the RD pattern, and the second eQTL is far away from the TSS. And are located, if you look at chromatin states, there are some enhancer regions around this peak. And this peak will locate in the same TAD region with their target genes. So the next step, we start to look at if these resources we developed, the GTEx resources can really help us to understand the genetics of complex traits in pigs. So we start to integrate it with these QTLs, integrate these QTLs with the GWAS summary statistics from 268 complex traits of very important economic value. So there are over 220 students, postdocs got involved in this analysis and data preparation. And this is a very busy panel, but there's a very good evidence to show that these eQTLs are very important to understand the genetics of the complex traits in pigs. So let's start with the first panel, the A panel. This is the sample enrichment. So we look at if the enrichment, if these QTLs are significantly enriched in the GWAS loci. And in general, you can see this significant larger than 1. And if you look at the difference in enrichment, and you will see the eQTLs and the splicing QTLs can have a higher enrichment compared to other 3 QTLs. And we also do another analysis. This analysis just to measure the proportion of the total heritability that are measured by each of these QTLs. So, and then you can see the splicing QTL and the eQTLs and can contribute on average about half the heritability. And the C panel is just a focus on eQTLs, but we did the same analysis for other QTLs, and we will show you later. That's a focus on eQTLs. And so we did a list of these analyses, like co-localization analysis between eQTLs and GWAS loci, and also TWAS, single tissue TWAS and multi-tissue TWAS, and another analysis called SMR analysis. So that they have different basic assumptions. They are not totally overlapped with each other. But in general, you can see about over 60% of the GWAS loci can be interpreted by eQTLs detected by at least one of these methods. And then we start to ask if the co-localized genes with the GWAS loci, are they more likely to be the nearest genes of that particular loci or not? Here, if you look at this bar, these 2 bars, and in general, we'll see the larger proportion of the co-localized genes are not the nearest genes of that GWAS loci. And the E panel also show that, so we separated the GWAS based on the significant level from the less significant to the very strong significant. And then you will start to see this co-localization events and the proportion will increase. So the GWAS with more evidence have more likely to be co-localized with eQTLs. And the F panel, we start to look at the contribution of the eQTLs or E genes. If they are detected in multiple tissues or more likely to tissue specific, they have different contribution for the GWAS co-localization. So this is just the average across all the 268 complex traits, and that traits including different kinds of the complex traits. So likely, there are almost all the tissues that got involved in this complex traits. And then as expected, you can say if the eQTL are more likely to share across all the tissues, they have a higher colocalization events with the GWAS. But for each particular traits, complex traits, they may be different because you need to detect the most relevant tissue for that particular and complex traits. And then we start to ask this question. So the G panel, so in general, we can say this is a sample size. This is just the sample size for eQTL discovery population. So in general, we can say if sorry.
Continue.
Okay. So the sample size increases can in the eQTLs can give you a higher power for the colocalization events. And then we need to, if you want to detect the most relevant tissue, you need to adjust it for this sample size. So after adjusted sample size, we take some complex traits as example. So here, the first trait is called the average daily gain, and we found the most relevant tissue is the elin, and followed by brain, I think, but I didn't point here. And for the back fat thickness, the most relevant is adipose. And for fertility traits, the most relevant is uterus. So this looks like a biological relevance. And the eye panel is just a very interesting result I would like to point here. Is so based on the TWAS results, we found this ABCD4 gene is the top association in whose expression in the small intestine was associated with average back fat thickness. And if we focus on the genes and look at the TWAS result across other tissues where we have information, and we also can find brain and have a very good association. But if you look at this gene in human, you also found that gene expression in brain and the intestine are associated with BMI or weight in humans. And by looking at the colocalization, so this panel is from the GWAS, the Manhattan plot for the GWAS, and this is from eQTLs, the eQTL from brain and small intestine. So this data are generated in different populations, totally different. This is Duraka, this is the multiple rates, but you can see a very perfect, beautiful colocalization. And there are two snails were colocalized, and these two snails have a good LD between each other. And if we focus on these two snails and look at the chromatin states around them, you only can see enhancers in the gut tissue and the brain, and no enhancers in other tissues. And as I said, we did the same analysis for splicing individual exon and link RNA genes. At the end, we put all these QTL together. So we found that this QTL are not exactly overlapped with each other. Each of them can provide additional information for understanding the GWAS. Like some of the unique or colocalization were only found between the average gene expression and GWAS loci, and also the splicing. So, but at the end, there are about 80% of the GWAS loci can be interpreted by this for different kinds of QTLs. And this is just some example. So the left panel is just to show this is the cis-eQTL specific colocalization between GWAS loci. So that is the target traits, the average daily gain, and you can see this the tissue and the gene name, the average gene expression, or have a very good colocalization, but not for splicing and any individual exon. But this is the splicing QTL specific colocalization between GWAS loci. So the GWAS loci, the GWAS is average the back fast thickness, and we didn't see any signal for the average expression for this gene in small intestine, but a perfect colocalization for his splicing patterns. And so here is just some specific colocalization between exon QTLs and the particular exon QTLs and with the GWAS loci. And we also look at the enhancer QTLs, and also we also detect if any SNPs can influence the linker expression, and then they target the protein coding genes and the GWAS. So I do find hundreds of these examples. So in general, from here you can see these results are very good. It's a product of very good resources and to understand the GWAS, GWAS loci, or maybe understand the genetics of the complex traits in pigs. And the last part of this project, I started to compare the pig GTEx with human GTEx. I just want to show if the pig are really a potential biomedical model for some particular GWAS, the complex traits of diseases in humans. So the first comparison, we start to look at the tissue-specific gene expression and also their regulation. So the A panel is just shows the top value. This is a statistics to measure the tissue-specific, the tissue specificity of the particular gene. And we calculate it in pigs and the humans separately. But for the ontologous genes, you can see there are good correlation with each other. And that also means the tissue-specific expression, the molecular phenotype is conserved between the two species. And then we look at the E genes. If the E genes detected in the same tissue have more overlap with each other. And in general, you can see the red dot is a matched tissue. That's the enrichment of the E genes detected in muscle in pig, and with that E gene detected in muscle in humans. So in general, you can see the red dots were located in the top level. But you need to keep in mind that the sample size for eQTL detection is totally different between the two species. But we still can see relatively enrichment. That's also indicated the colocalization, the conservation between these eQTLs. And then we also simply to look at some variants to see if they have the same effects. But we do see some, but but we didn't see a very good example actually. The eQTLs, I think the good way that we need to compare these eQTLs based on the matched sample size between the two species later. And another interesting analysis to look at the conservation of the complex traits genetics. That's all based on the TWAS results. So we take the about actually over 150 complex traits in UK Biobank or other consortium, and then we do the TWAS, and for each of these complex traits across all the tissues in human, and exactly the same did in these 268 complex traits in all the tissues in pigs. And then we start to look at the correlation between the effect size from the TWAS, also within the ontologous genes, in the matched tissue. And then we just plotted here, and the colors were separated about different the trait domains from a human, and this is and this will indicate, this just indicated the p-value was adjusted by Bufferonian correction. So we can see a lot of the significant correlation between the orthologous genes in terms of the file size on the similar traits. And just one example, like in brain, we found the top correlation and the conservation between human height and the days. This just means the how many days it take for the pig to grow up in the 30 weeks, like 100 kilograms. And also in the brain, we can see the BMI has a very significant related to the days, and all the other things were founded. And we also found by looking at literatures, we found that the findings probably relevant. And so these two panels are just two examples. Like in brain, we can see the BMI and the days in the BMI in human and the days in pigs. The gene expression in brain have a low but a significant correlation. And the red dot is just means the both significant in the two species. And this is another trace. So at the end, a short summary about this pig GTEx. So we generated the largest expression atlas in pigs, so that including over 100 tissue and cell lines. And this data can be used to better functionally annotate the gene function, the genes in the current pig genome. And another resource is we call the parallel phase for the pig GTEx is by generating the cis-eQTLs for 5 different kind of molecular phenotypes. But besides this, we also detect the QTLs for co-expression networks across all the tissues. And we also apply this resources to understand the complex trees and adaptive evolution. So I didn't show the results from this part, but we do find a very interesting findings here. And then by comparing these two results between human and pigs, so at different levels, gene expression and their regulation, the genetic regulation and TWAS, we can show that the pig probably a very good potential biomedical model for certain complex traits of variants or genes. At the end, we will make all this data publicly available by building an open access portal. So at the end, I would like to thank all the other collaborators around the world from this institute, and also some colleagues from MRC and also Rosalind, Albert, and Paul. And thank you very much. This is all I would like to share today. Yeah.
Thank you very much, Lingzhao, for this very extensive piece of work that you've presented. I don't even know where to start or to begin. It's so much information. Yeah. Thank you very much. Very good job. Yeah, so we haven't received any questions yet, but maybe I can start with the first one. So you say that you have lots of public data that you sort of combine into this one analysis. What are the challenges, and how do you solve the issues when you have data that were produced from different sources? How do you manage to combine them, and what are the steps that you take to sort of make them?
Yeah, and so actually, because the data are generated at different places, and there are lots of metadata information missing. But after the analyzing all this data, and we first will do some cluster, and we also start to look at the data quality and how they are related to gene expression, for example. And we will remove the low quality data size, and based on the cluster, we remove some obviously outliers. So they were labeled as maybe as liver, but totally clustered far away from liver. So after different kind of data filtering, and we got some data sets we think is a good data size and can replicate it. can represent the tissue, and then we use it for the downstream analysis. So it's a very time consuming to get all the data down and to look at each individual publications, because we need a lot of information missing. We need to go to their publications and read them from the methods and to retrieve this. So, and also, by some confounding factors, we need to training some methods. Yeah. So training the methods and to go to the states, like unknown factors and remove like a cell type composition, some others on sex and ages. Yeah.
Okay, we received one question from James Prendergast. So he says, nice presentation. If you're calling 3 million variants, have you estimated what proportion of all variants in the samples you think this is, and how does this impact the fine mapping?
Yeah, that's a very good question. Yeah, because in the reference panel, we do have over 14 million SNPs, but the RNA-seq, we only can call the SNP from RNA-seq data, and that SNP was just unevenly distributed or only focused on the transcript region. So actually we didn't estimate the problem. This is all variants in the samples. I think that maybe just about 10% of the SNPs in all the samples if we base the reference panel. But if we consider the other SNPs, we do not do a very good imputation. So that we just remove that, especially for the SNPs that are far away from the transcript region. And another is this and how the impact? Yes, I think so. So this because we are losing some SNPs, and I think that may impact the fine mapping. But here, if we just focus on cis-eQTLs, the region, and just 1 megabase around the TSS region, we look at the SNPs, the distribution. But we found that it's not that a big problem if we just focus on the cis-eQTLs within 1 megabase. But if we start to look at the trans-eQTLs, that's a big problem for this. Yeah. Yeah, they do have some impacts. Yeah. If you have the whole genome sequence, the deeply whole genome sequence, you can start to look at the rare variants, and you may find more SNPs. And we also compared with the human. So we found that the fine map of the SNP is a little far away from the TSS compared to the human fine map of the SNPs to the TSS. I think that's probably the imputation problem. But this is a very good question.
Sorry. So I need to unmute myself. Wait.
Yeah. I think that hi, Julie. Yeah. Okay, very strong background.
Okay, so sorry. Yeah, I think now. Yeah. Okay. Sorry, we're sitting in the same room.
Okay.
Okay, it's not doubling anymore. Perfect. I just have a really quick question. So is there an association between the gene length and the number of cis-eQTL? And if so, do you have to account for this? So for example, the longer the gene, the higher the likelihood that you have an eQTL located in it?
I'm sorry, I got some noise here. I close my window. I'm sorry. Could you repeat your question again? Yeah.
Or just type it. So I said, is there an association between the length of a gene and the number of cis-eQTL? So for example, the longer a gene is, the higher the chance that you have an eQTL located in it. And if so, do you have to account for this somehow in some of your subsequent analysis?
Okay, so you mean that if the longer gene more likely to be detected, or if the gene have a higher expression, are more likely to be detected eQTL. We checked that, we didn't find any correlation.
Okay,
Yeah.
perfect.
Yeah, that's we later, I think in 2 months you will see the preprint and all this cost. We did a lot of analysis for that part and to look at if the eQTLs are directly related to the gene expression. Because if a gene with a high expression, they more have a high, the phenotypes are high with a high accuracy. So they're probably more likely, but we didn't find clearly evidence for that.
Okay, thank you.
Thank you. I think we'll slowly finish this call, and thank you. I'll ask you a quick question before you go. If you had to summarize your presentation in a sentence, what is the takeaway message, and what is the direct application of this work that you've done?
Yeah, I think that's a good thing. So I think that if just one or two questions, I think there's a molecular phenotypes to can help us to understand the complex traits. And I think the more relevant data probably need to be generated, and also a lot of the tissue with a very small sample size. We need to encourage people who are interested in this molecular QTL mapping and to apply relevant grounds based on these results. And so we are very happy if anyone would like to use this data size, and to apply relevant grounds and hope this can be used as the primary results. So so the most important that the molecular QTLs maybe measures for understand the complex traits.
Thank you very much.
Thank you very much, and very happy to share this.
Very nice work. Thank you.
Thank you. Bye. Bye.
Bye.
Yeah.